So DNA replication in the cell cycle is actually one of the cycles that I like in biology because it's quite simple.
So firstly there is mitosis,
which is split into 4 other stages of prophase, metaphase, anaphase and telophase.
Next 3 stages are part of something called interphase, the first of which being G1, which is growth phase. This is when sort of all the proteins and everything duplicates in preparation for S phase, which is where DNA replication occurs. And the next one is G2, which is secondary growth and checking over of the newly synthesized DNA.
So as DNA replication is semi-conservative, it means that each new DNA strand contains one new strand and one old strand, or one template strand. This means that the initial DNA has to be unwound. This is done by an enzyme called DNA helicase, which is the little triangle here. DNA helicase breaks the hydrogen bonds between the nucleotides on the 2 strands of DNA. Well, it's one strand of DNA made up of 2, if you get what I mean. But because of DNA's helix structure, this can cause some stress on the actual helix. So an enzyme called topomerase sort of nicks the DNA to assist in the unwinding by DNA helicase. If the DNA strands can't rotate, then the stress will build up and it could cause damage. The next stage involves an RNA primer being added by DNA primase. This allows DNA polymerase to add complementary nucleotides to the original template DNA strand. This occurs on both sides of the now exposed DNA, and the polymerase moves from 3 to 5' in order to build the new strand 5 to 3'. It has to do this because DNA polymerase can only add nucleotides to the third carbon on the ribose sugar ring of the nucleotides. So just remember, DNA is read 3 to 5', but DNA is synthesized 5 to 3'. So in other words, the original template strand is read 3 to 5, and the new strand that's being added on is done 5 to 3. So you'll have noticed here that I've labelled the 2 different sides, leading strand and lagging strand. This is because DNA replication is continuous on the leading strand because it's sort of like moving along towards in the direction of DNA helicase. But on the lagging strand, DNA replication is discontinuous because obviously the RNA primer has to keep like sort of being added on further down so it can keep building backwards from it. The diagram works better than how I'm going to explain it, but this is discontinuous, so the fragments that are made between each RNA are called Okazaki fragments, and they're about 100 to 500 nucleotides long. But you're probably wondering, how does this replication start in the first place? And well, it occurs in something called a replication bubble. So there's a local opening of the helix, and then the RNA primers are added, and then sort of the whole process that I've just described occurs. Where this opens, it creates a fork so that the 2 primers and the helicase can sort of like move along. It's much more common in eukaryotes to have a replication bubble, whereas in bacteria it's not very common, as there's only usually 1 bubble per DNA strand, and the strand can actually be duplicated in around 30 minutes, whereas eukaryotes, which are us humans, can have like 30 to 50,000 per cell division. This is because we have a lot more DNA, so if we only had 1, like a bacteria, it would actually take around 35 days for 1 cell division replication.
And then here's just a little summary table of some of the enzymes that I mentioned earlier.